Hello again. We're going to do Chapter 11, PowerPoint, and I hesitate to say this is going to be quick because every time I say quick, it takes a while. But I just want to, there's a few fundamental things about chromosome structure that we're going to talk about here. And so this chapter covers not only basic chromosome structure, but it also talks about organelle DNA. And we're not going to talk a lot about organelle DNA in this course, just because we can't cover everything. But as you know, I'll just draw it right on this slide. So there's a cell, there's a mitochondria, there's a nucleus, and inside the mitochondria, there's a little circular chromosome. So at this point, all you really need to know is that mitochondria, and maybe this is a plant cell and it has a chloroplast. Just know that these, that looks like a little face, but these organelles, mitochondria and chloroplast, have their own DNA. So that's really all you need to know at this point. We could certainly go into that in more detail, but right now we just want to talk about chromosome structure. All right. So chromosome structure, the idea is, so sort of that we're thinking about chromosome structure. We need to think about so the DNA and how that DNA gets packed into the cell. So let's say that we have a cell and in a nucleus. So there's a nucleus of a cell. And that's probably 10,000 times bigger than a real nucleus looks like. So a nucleus is a really small thing, maybe 1 micrometer, so 1 millionth of a meter. So it's pretty small. Then you got your DNA, and the DNA is organized into chromosomes, and in humans we've got 23 pairs of chromosomes. We know a little bit about DNA structure. If we just talked about DNA structure, if we've got a double helix, we've got base pairs, we know that between a base pair, there's 0.34 nanometers of DNA. So there's 0.34 nanometers per each base of DNA. So if we just have like A, G, C, T, on and on, the distance between this is 0.34 nanometers. All right. But if we take all the chromosomes together in one cell, there's 3 billion. So that's 3 billion with a B base pairs. And there's 0.34 nanometers per base pair. So with that, so if we take 0.34 times 3, that's essentially 1 times 10 to the 9th nanometers. And that actually equals 1 meter. So we can't really draw a meter, but let's say that's our DNA. That's 1 meter. If we kind of linked it all together, we've got to take that and get it into a 1 micrometer nucleus. That means we have to condense this DNA on the order of 1 million times. That's because a meter is 1 million times bigger than a micrometer. So we need to scrunch this DNA down, and the scrunching is called condensing the DNA. So this is what we call the DNA packaging problem, because we're packaging a lot of DNA into a small space. And I think the late, great Robin Williams can explain this better than I can. So let's take a quick look at that.
It's all part and parcel of the whole genie gig. Phenomenal cosmic powers!
And now we've tried to figure out how to get out of here. get out of this thing. It's always something with this. Exit full screen. There we go. Close out of here. All right. So what I was trying to show there is so what I was trying to show there is DNA, which has a lot of information. 1 billion, well actually it's 3 billion base pairs. And this DNA codes for everything that is present in an organism, in a human. So all the features of a complex organism, all that information is contained in the DNA. So we'll call that phenomenal cosmic power. All right, or phenomenal genetic power at least. But we got to get it down into a nucleus, and that's 1 micrometer. So that's really small. So that's an itty bitty living space. So how do we package a huge amount of DNA into an itty bitty living space? So we call this the packaging problem, the DNA packaging problem. All right, and so that's where chromatin structure or chromosome structure comes in. All right, so there's a couple things. One thing that happens with DNA is that it gets supercoiled. I don't necessarily talk a lot about supercoiling. It's not that big of a deal. But the DNA gets twisted, and the enzymes that twist the DNA are called topoisomerases. So just see a picture. You've got a circular DNA, and the DNA might not necessarily be circular, but it might be connected to like some kind of framework. The DNA kind of loops like this, and then you can twist this DNA, and as you twist it, it gets more compact. So this process of supercoiling is one mechanism for compacting DNA. That's not the main thing we're going to talk about. Now, first we'll start with bacterial genomes. That's a little bit of a different problem. Bacterium is probably about as big as a nucleus, but bacterium has a bacterium itself is just a circle. So let's say this is E. coli, or it's an oval or whatever, and it's got 1 chromosome, and it's a circle. And then when that cell divides, in order for it to divide, that DNA has to replicate, and then the cell divides. So this is just bacterial. This is what a bacterial chromosome looks like. This chromosome is about 1 million base pairs. So it's 1,000 times smaller than a human genome. And it has to get packaged into the bacterium. But the way it's not as complicated as it is in like a human cell. So there is some packaging that has to be done. So if here's a picture of E. coli. I always have to remember to grab my pen when I switch sides. So that's E. coli cell. And this is all the DNA that gets released from the cell. So if you just kind of break open the membrane, you can release all this DNA. It's pretty cool. But if you got to look at this, you can tell that this DNA is kind of in a loop. If you follow that all the way around, it kind of comes back to a loop. And so the idea is that there's these some kind of proteins in the cell, and the DNA is looped into these proteins. We'll see a similar kind of structure in eukaryotic cells, but this is basically it for eukaryotes. We got for prokaryotes. So these are prokaryotes. The idea here is that the DNA is packaged into the cell, but it's going to be a different different mechanism than in mammalian cells. And so this is you might twist this and supercoil it, so you can have supercoiling just to help kind of pack it in there. And there's probably other proteins, and I'm certain there are other proteins that are associated with this DNA that just aren't shown here. But it's not going to be the histones and that sort of thing that we see with eukaryotes. So we're really most interested in eukaryotic chromosome structure. And an important term here is chromatin. So chromatin equals DNA plus specific proteins. Mostly these proteins are histones, so that's the main type of protein that we're going to talk about. So these are DNA binding proteins that make up chromatin. So chromatin is a very specific type of material. If you have a cell with a nucleus and with chromosomes, the chromosomes are made up of chromatin. And so we'll talk about some experiments, and it's important what you isolate from the cell. If you're isolating chromatin, that's different than isolating DNA. DNA is called naked DNA, which is this DNA without any proteins, and you can get that. You can isolate DNA and get rid of all the proteins, but then you have just DNA. A lot of times you want to isolate chromatin and study what's going on with that. So chromatin, we can think of it in terms of euchromatin and heterochromatin. And if you think of a chromosome, and we've drawn chromosomes like this before, this region is the centromere, and it's very dense. And when you see it, it's dense under the microscope. And this is heterochromatin. And then the regions of the chromosome that are sort of the main part of the chromosome is called euchromatin. And then we also have telomeres that are at the end of the chromosome, and that's also heterochromatin. So euchromatin is where all the genes are. So if we do this sideways, we had some euchromatin. This is where your genes are going to be located, and they might be expressed. So this is where all your genetic loci are. Maybe you have an A gene, a little B, a big C. So the genes tend to be located in euchromatin. It's not 100% true. Nothing's 100% true. But that's the general idea. And then heterochromatin is usually structural elements of the chromosome, although sometimes you can have some genes in there. So we need to talk about the structure of these things and the role of the histones in forming these structures. So these are just some general characteristics of euchromatin and heterochromatin. Euchromatin is less condensed, heterochromatin is more condensed. As we'll see as we go and look at the structure of chromosomes, the DNA is wrapped around histones, so we'll see our DNA wrapped around these structures called nucleosomes, but we'll come back to that in a minute. And the heterochromatin is more condensed, so these nucleosomes are squished up more. So we'll see that picture a lot. Sometimes we can go back and forth between these two states. So location, so chromosome arms, that means if you've got, just we drew before, we have the centromere, and this would be the arm. And maybe in eukaryotic chromosome, you might have a P arm. I guess this one looks shorter, so that would be the P arm and the Q arm or something like that. And then when we look at chromosomes, there's the euchromatin basically has what they call unique sequences. It's basically just kind of different sequences, the different genes have their own sequence and they code for their own proteins and stuff. Then we have repeated sequences. That's common to centromeres and things like that. So if you look at the sequence of the centromere, you'd have the same sequence repeated over and over again. And that tends to be the kind of sequence that forms heterochromatin. Most genes are in euchromatin. There are a few genes in heterochromatin. And the heterochromatins replicate a little bit later. And their transcription, there's that most of the transcription occurs in euchromatin, and crossing over also happens in euchromatin. And the idea for that is that because this is all condensed in the heterochromatin, the euchromatin is more accessible to things. And that's a big theme. So euchromatin is more accessible. That means if proteins have to get in there and transcribe the genes, the euchromatin is more open and looser so that it can be accessed by these different proteins. And this is less accessible. So here's a protein, tries to get in and transcribe a gene, and it can't get in there because it's too scrunched up. So we have to think about nucleus chromatin structure, and these are the different types of structures that we're going to talk about. And we have the nucleosome itself, and the nucleosome is going to be all right. So when we're talking about chromatin structure, we're always going to be talking about. So it's this word chromatin. Chromatin is going to be DNA plus proteins, and those proteins that we're going to talk about are called histones. So the nucleosome has 8 histones, and we can kind of think of it as drawn like this, where there's 4 histones, and then there's actually 2 of these structures, and it's kind of like a yo-yo. If you look at it from the side, we got one loop like this, and one loop like this. We got 4 histones here, and 4 histones here. So that's what that thing's called, and it's called the octama, because there's 8 histones altogether. So it's the histone octama. And then there's DNA that's wrapped around it, and it's about 146 base pairs of DNA, and that is called your nucleosome. Now the chromosome is when we have a bunch of nucleosomes together, and we've got DNA wrapped around there. There's these 8 histones, and I'll tell you what those things are in a minute. This is linker DNA. And then the chromosome, there's another protein called histone H1 that's there, and that's called the linker histone. So that's histone H1. And this DNA containing, so this whole structure consisting of histone H1 is called the chromosome. And then we're going to see, so this is called the 10 nanometer fiber, just because it's 10 nanometers in diameter. If you look at it under the electron microscope, it looks like beads on a string, and so a lot of times we call that the beads on a string structure. And then this gets further condensed, so there's your beads on a string. Then we're going to further condense that into a more condensed structure like this, and that's 30 nanometers in diameter, and that's called the 30 nanometer fiber. And then we further condense this into these more complex and more condensed structures. But that's kind of the overview of where we're going with this, but we'll look at each thing specifically. So here's, we think of this DNA when we sort of monitor the progress of compacting the DNA into the nucleus. Keep in mind this is our phenomenal cosmic power itty bitty living space, so that's what we're trying to do, is scrunch the DNA up. These pictures are always a little bit weird because the way we sort of monitor the state of how the chromosome is condensing, and so this is called chromosome condensation. And we go from a less condensed to a more condensed, is that the chromosome gets thicker. And so this is, by the end, we'll have a 1400 nanometer chromosome, which is way thicker than where we're starting, but they always show the DNA as being wider on this screen in the picture. So that's a little weird. But reset this. So it's good now. But anyway, so your DNA is 2 nanometers, and then when you scrunch it up, you get to 10 nanometers, and now somewhere along the way, that 10 nanometer fiber has been called the 11 nanometer fiber. So somewhere they measured it more accurately. Call it 10 nanometer, 11 nanometer, whatever you want to call it. I'm sure it varies. I'm sure in some places it's 10 nanometers, and some places it's 11. Everything's, anyways. So then we've got our nucleosome, is this thing. And it looks like there's a little globe in this picture, but there's 8 histones. So the 8 histones are the histone octamer. And with the DNA wrapped around it, it's the nucleosome. So this is the nucleosome with 8 histone molecules and some DNA wrapped around it. And then the chromatosome would be this whole thing is the chromatosome. And that has about 200 base pairs of DNA included. All right, so and then you got your histone H1. That's a linker. It kind of binds to the DNA in between nucleosomes. And then this gets further scrunched up into what we call the 30 nanometer fiber. And then this here is the 30 nanometer fiber that's getting further scrunched along, and this attaches to something in the nucleus, and that's called the nuclear scaffold or matrix. There actually used to be really competing scientific groups, and one group always called it the scaffold, and another group always called it the matrix, and I'm not sure who ended up there. So you got this nuclear scaffold, and the 30 nanometer fiber attaches to it and forms these loops, and then we get further scrunching, for lack of a better term. Officially, scrunching is called condensation. But what we really do is we think more about what's going on here. At least conceptually, we're going to talk a lot about going back and forth between these states of chromatin, where the 10 nanometer fiber or 11 nanometer fiber is less dense than the 30 nanometer fiber. And the idea is that in interphase, the chromatin is more spread out, and then the chromosomes that we were able to see during metaphase look like that. So that should look familiar to you as a metaphase chromosome. All right, so we're going to focus mainly on this sort of area right here. All right, so here's all that stuff with these little balloons that the PowerPoints have, which to me it's a little bit busy, but at least it has all the information there if you need it. So more information, good, but it gets a little bit crowded. All right, so we're not going to worry too much about what happens after the 30 nanometer level. So let me just back up here and put in a new slide so I can draw this in a sense, in a way that where you might draw it. And so we have a 10 nanometer fiber. which consists of a core nucleosome. So here's our histone octamer. And the histone octamer has 8 proteins, and they're all histones. And they're called H2A, H2B, H3, and H4. And there's 2 of each of these. So you might see H2A, H2B, H3, and H4. And then there's a second version of this, kind of right behind it. And then your DNA kind of wraps around there like this. And then if you have your DNA coming along here, and you've got another histone octamer, and your DNA wraps around that, and then that, you get a whole bunch of those together, that's your 10 nanometer fiber. Then your 30 nanometer fiber is you take these things, these nucleosomes, so it's a nucleosome, and it gets further condensed, where, think of this 10 nanometer fiber as a big long hose or something, like a garden hose, and then you coil it up like that. And so if you take that hose and you look at it from the side, it sort of looks like this. And if you turn it around and look at it going down the end, it kind of looks like that. And so each loop of this coil has 6 nucleosomes in it. So that extra U in there. So that's 6 nucleosomes in each coil. So when you look down it, and then you've got your DNAs also in there as well. So your DNA is kind of wrapped around all of these things. And so if you look at it going down the long way, there's a whole bunch of these stacked kind of coming out at you, out of the screen, that have 6 nucleosomes per coil. If you look at that on the side, you can only see 3 of those nucleosomes. And we know that each nucleosome is about 10 nanometers. So when you look at the side, it's about 30 nanometers. So that's the 30 nanometer fiber. And again, we're going to kind of go back and forth between these two states, the 10 nanometer fiber and the 30 nanometer fiber. So if you when people study nucleosomes, one thing you have to do is you have to isolate chromatin. And then a lot of techniques that they use to study chromatin is using an enzyme called a nuclease. And nucleases cut nucleic acids. So in this case, DNA. So it can be called a nuclease, or sometimes it can be called DNase. So nuclease here is also called DNase. And it cuts the DNA, but it only cuts the DNA if it's not bound up by proteins. So in this case, we've got some free DNA that's exposed in between the nucleosomes, and if we cut it with a nuclease, we can release individual nucleosomes, and that's how they came up with what is in one of these nucleosomes. So if you treat chromatin, notice we start with chromatin, not DNA, not just DNA, and we treat it with a nuclease, we can release a nucleosome, and then when they looked at the proteins, they found that there's 8 proteins in there, and there's about 146 base pairs of DNA. Now here in this view, it's showing some of the different proteins, and here the yellow one is H2A, and then there's the other H2A, where there's 2 of each, and then here's H2B, and here's the other H2B, so they're calling it one H2B and one H2B prime, And then here's H3 and here's H4. And there should be two of those, but you can't quite see them. So that's just one view of the nucleus. The white is the DNA, and the different colors are showing where those proteins are. I've seen better views of the nucleosome before. You can look it up and find better ones. But that's the idea. That you've got that sort of nucleosome. I like to think of it. So you've got these 8 proteins. And then you've got DNA wrapped around it. Now, one thing we'll notice about these histones as we go along is that there's all these different histones, and then so this is a protein, that's a histone, and then each of these histones has these tails. So the histone tails kind of stick out from the structure, so you can kind of see a tail here, maybe a tail here, and these tails can get modified. And the modification of those histone tails can affect how condensed the chromatin is. So that's a big theme we'll come back to. Just a close-up of what we just looked at. And there's a picture of very much more close-up of that. All right, so let's bail out of this for a second, and let's go to let's do a quick little Wikipedia thing. All right. See if that gets us in. Oops. So there's lots of different views of it, just to give you an idea of what we're dealing with. So lots of different ways to think about it. Here's a good one over here, where you've got H2A, H2B, H3, H4, DNA wrapped around it, and then you've got your H1 thing there. So that's probably kind of a good one. Down here you've got a nice picture showing the 6 nucleosomes that are in a ring of the 30 nanometer fiber. So you can look at whatever views you like. That one's kind of cool. It's got the DNA wrapped around the outside, and then you've got your different histones that are in different colors. So histone's pretty cool structure. Feel free to look around and find some videos that give you a better view of it. But as far as you're concerned, you have to draw it out in kind of an abstract form. All right, so chromatin structure, we just talked about that, and then now we want to relate chromatin structure to gene expression. Really, the last section of this course, after this next exam, is going to be all about gene expression, and we're going to go into this in much more detail. But right now, I just want to give you a preview of that to kind of give you an idea of why chromatin structure is important. And since they cover that in this chapter, so we might as well go over it. So chromatin structure is dynamic. It changes a lot, and when it's condensed, the genes can't really be expressed, and then when they're not condensed, when they get loosened up, then you can express the genes. So we're going to look at a dramatic example of this in these things called polytene chromosomes, and look at some chromosomal puffs, and then we're going to look at a DNA sensitivity experiment that demonstrates this. This is a little bit obscure in this PowerPoint, but I think we can get to it. And then we're going to relate this to this concept of epigenetic changes in gene expression. So that will take you to these things in turn. All right, so this is a special chromosome in Drosophila called a polytene chromosome. And polytene chromosomes are large chromosomes that you can see under the microscope. It's kind of fun to talk about where they come from. So here's a little larva. Not a very good picture of a larva, but there's a larva. So this is a Drosophila larva. And this is its head. And when these larvae are cruising around, all they're doing is eating. They got some mouth parts up in here, and they got some salivary glands, and they can actually digest things with these salivary glands. And if you rip off the head, so you take your larva and you rip off the head, use some forceps, these salivary glands come out, and they kind of look like that. And I've actually done this quite a bit. And then in there's these giant cells, and you squish these things on a microscope slide under a cover slip, and when you squish them, these chromosomes get released out of these cells, and you can see them under the microscope. And this is, excuse me, I believe this looks like it's the X chromosome, just from the numbering I can tell that. And so this is a Drosophila. And you can see, when you look at these chromosomes, you can see these different bands. And so there's different chromatin structure. When you stain these chromosomes, you can see different regions of the chromosome sort of stick out and band differently. It's kind of cool. You can actually look at these things, and you might look and see that there's a chromosome deletion. And maybe this big band is missing, and you can tell that. So let's say we looked at a chromosome and that was missing. But another thing you can look at is something called chromosome puffing. And so here's this region. So this is kind of kind of a farther away view. But the idea, so this is between B4 and C1. So we're kind of looking at, it's hard to say exactly. But the idea is that we're looking at basically this region, and then when we look at this, it's much more expanded. And so this region's much more expanded. And so we call that a chromosome puff. So we have our normal chromosome, and then it's sort of puffed out like this, and it's looser. So this is loose. And the idea is that the genes in this region are off under this circumstance and on. So the genes are off and on. So it might be a gene, and so what we're talking about is transcription. So when a gene is turned on, it's transcribed. So we got a high level of transcription, and you can tell that, and so you can visually see that in the region of high transcription. Now you'd have to do an experiment to show that there's a lot of RNA there, and then this is low transcription. But this is visual evidence that transcription correlates with a change in chromatin structure. So this chromatin puff, or this chromosome puff, is an example of a looser structure, and that correlates with transcription. So we're not showing the transcription here, just take my word for it. So another way to correlate chromosome structure with transcription is to do what's called a DNA hypersensitivity experiment. All right, so now pay attention, because this historically has been difficult for students to kind of grasp, and so it might take a little work for you to think about it. So first of all, this is just reviewing what the structure of a nucleosome is. You've got histones, and you've got these tails that are sticking out that are available for different kinds of modifications. These are some of the modifications that we can see, and what these do is they control how tightly condensed the chromatin is, and how tightly condensed it is determines its function, meaning is it transcribed or is it not. And the nucleosome is the basic packaging unit of the DNA, and it's around this histone octet or the histone octamer. So we just talked about that. So it looks like this. Oh, that's cool. I like that picture. It's got some colors. So that's what we're talking about. And this is showing where a gene might be located relative to some chromosomes, to a chromosome. So let's back up a little bit. Let's see here. So let's see, I just want to have a blank slide. So I want to review gene structure a little bit. So a gene made out of DNA, make this a little bit longer. This is a gene. This is the transcription start site. Alright, so I'm definitely not sticking to this being short, but yeah, sometimes it's worth taking extra time. We have a transcription start site, or a TSS. And if we're going to transcribe this gene, we need RNA polymerase. And we're going to talk about this more in the next chapter, but we need RNA polymerase to transcribe this gene. And the way that RNA polymerase finds that starting point is there are sequences near the gene called promoters, and then enhancers, which can be farther away. And there are other proteins that bind onto these enhancers, and there's proteins that bind onto the promoter, and all of these proteins have to bind in order to transcribe the gene. So the thinking is that if we've got DNA, so I guess before I had, we got our DNA wrapped around these nucleosomes, and we have a gene in here. In order for these other proteins to bind onto the enhancers or to bind onto the promoter, and then to get RNA polymerase to get in there, we need this chromatin to be opened up like this. So we need to have the chromatin loose so that these proteins can find the DNA that they need to bind and bind it. And then once you do that, then you can transcribe the gene. But if your chromatin is all condensed, all these other proteins that need to bind so just different proteins with some different colors. So these proteins are trying to bind and trying to find their gene, and they can't get in there because the chromatin is so condensed that these proteins can't find the DNA that they need to bind. So in order to turn the gene on, you need to loosen it up. And then these proteins can bind. So they can find their spot. They can bind. We get a blue one down here somewhere. A black one somewhere. And then we can transcribe the gene. So this is the 30 nanometer fiber. It's condensed. And this is the 10 nanometer fiber. And it's decondensed. And this is inaccessible to proteins binding it. And this is accessible. Now, one of we have these tails that stick off the chromatin. So these are nucleosomes, and different modifications of those tails allows it to be either loose or tight. So I'll just mention one type of. Let's find some crazy colors for this. So let's say we've got, there's a site here that can be modified, and we'll just talk about one modification. There's something called an acetyl group that can get added to these tails, and that acetyl group causes the chromatin to loosen up. So if we have what's called acetylation, and these are acetyl groups, then the chromatin is loosened up. And if we have deacetylation, if we take those deacetylation, it takes those groups off and then it condenses the chromatin. That's the idea. We'll get into this a lot more in the last section when we talk about gene regulation. It's always good to give a general introduction. We need to talk about the enzymes that do this and how they know when to do it. But for now, I think. Alright, so that's kind of the overview. Loose chromatin, dense chromatin. When the chromatin's loose, then you can transcribe the gene. Simple enough. So that's what this is showing. Is we've got a region of dense chromatin, and we've got a region of loose chromatin, or condensed and decondensed. And how do we know this? So how can we ask the question of which part of the chromosome is condensed and which part of the chromosome is decondensed? So here this is showing there's some acetyl groups attached to the nucleosomes, and in that region the DNA is condensed. And so this is right here showing where transcription starts. And then these are some of the proteins that bind nearby. These are some proteins that bind farther away. And we're going to ask, we need to ask the question of how do we know that the chromatin is looser in these particular regions. So we're going to do an experiment called a DNA sensitivity experiment. And the regions that are more sensitive to DNase, which is an enzyme that chews up DNA, is going to be called DNase-1 hypersensitive site. And so that's what all these little arrows are showing sites where the DNase can cut DNA, and this DNase cannot bind and cut there. So DNase sensitive site, or DNase hypersensitive site, correlates with loose chromatin. Right, so DNase hypersensitive site equals loose chromatin. Then you'd have to do another experiment to show that this correlates with high transcription. So that's transcription. Alright, so let's take a look. So there's some decondensed chromatin and some condensed chromatin, and we're going to talk about how we can do an experiment to show that the loose chromatin is more sensitive to DNase. So the way this experiment works, so here they're showing there's some acetylation. On this chromatin, which makes it looser. And so there's an arrow here for acetylation going that way. And this is and then once it's loose, then transcription factors can bind and you can transcribe the gene. So saying the same thing again. Alright, so how do we do this experiment? The idea is we isolate chromatin. We're doing a DNase hypersensitivity experiment. There's an I in there, sensitivity. So DNase hypersensitivity. Isolate chromatin. Digest with DNase. Then isolate DNA. Notice DNA and chromatin are two different things in two different steps. Run a gel or do a southern blot. So a southern blot allows you to detect specific DNA. So if we run a gel, we'll see all the DNA from the cell will be present, and it will be all kinds of different sizes. If we do a southern blot, if DNA is present, only specific DNA will show up. So if we've got some DNA and we have some restriction enzymes that cut this DNA, and then we have what's called a probe that can hybridize to that DNA. When we do a southern blot, only this part of the DNA will show up on the southern blot. So don't get too hung up on the southern blot. Just know that the purpose of a southern blot is to detect a specific sequence of DNA. And so when we do this experiment, if that DNA is still present, it'll show up on the southern blot. But if it's gone, it won't show up. That makes sense, right? So let's say we do this experiment, and then we got different samples. And in this sample, no band shows up. That means that at this step, the DNA got chewed up. So that means that the DNase got in there and chopped up the DNA. So then when we did a southern blot, it wasn't there anymore. However, if the DNA was in a condensed chromatin, the DNase can't get to it. And so that DNA is still present. When we do a southern blot, it shows up as being present still present. So it hasn't been chewed up. So when it goes away or gets lighter, that's how we identify a hypersensitive site. So it's a DNase hypersensitive site. This site is more sensitive to being chopped up by DNase than the other site. Now, it might be the same DNA, and the critical thing is it's the same DNA, but it's in different tissues or different cells. So to show an example, and this is actually an example that I showed from another textbook for a long time, and they show the same thing in here, so it's good. So we have, so you isolate nuclei and isolate. So in the nuclei, there's chromatin. Treat it with DNase 1 to chop up any DNA. And then you cut it with a restriction enzyme, in this case BAMH1, run it on a gel, and then do a southern blot. And they're doing this with the globin gene, which makes hemoglobin. And erythroblasts are cells that are going to become red blood cells. And so these guys have high transcription of this gene. These guys have low transcription. So these are other cells that don't express this gene. And so this experiment showed that if you treat it with DNase, that this gene in these cells, same gene, different cells, is more susceptible to being chopped up by DNase, where these guys, DNase can't gain access because these guys are not transcribed, which means they're squished up into this structure. These guys are transcribed, so they're loose in this structure, and the DNase can get in there and chop up the DNA. So in these, the trick is that these are in different cells, same gene, different cells. So gene expression, changes in gene expression mean cells that have the same DNA express that DNA differently. And so this is what the result of this experiment looks like. So here we've got the southern blot that just allows us to detect the specific gene. This is increasing levels. So sometimes they'll show an increasing level like this. So increasing level of DNase. So we've isolated DNA, treated with more and more DNase. So that's what's going on over here in these 14-day erythroblasts. And as we add more DNase, we start to get rid of the DNA. So see this, where the DNA is going away? That means it's sensitive to DNase. Here, this is from those other cells where the gene is off, and the gene looks like this. It's scrunched up in condensed chromatin. DNase cannot access it, so it can't chop it up. So even if we add the highest amount of DNase, so that's 1.5 micrograms per mil, this gene is still, or the DNA is still present in those cells. So we're not in the cells. I mean, the DNA is present in both cells, but when you isolate the DNA and chop it up with DNase, this DNA doesn't get chopped up. And it doesn't get chopped up because it's condensed. So the whole point of this is that you can use DNase to tell you the state of chromatin in a particular gene in a particular cell type. So this gene, the beta globin gene, or the hemoglobin gene, is correlated or associated with decondensed chromatin in erythroblasts, where it's highly transcribed, and it's associated with condensed chromatin in these other cells, these MSB cells, that are not transcribed. So this is a confusing diagram that is showing, so now these little red arrows are just pointing to a region that is a hypersensitive site. Okay, and so these are different cells. This is the developing chicken embryo, and at different stages of development, a different hemoglobin gene is expressed. So early on in development, this gene is expressed, and then in the adult, these genes are expressed. And all this is saying is that if we look at hypersensitive sites by doing the same experiment, and we ask which regions of this DNA are sensitive to DNase at different times, that early, so here, this region is sensitive because it's got an arrow, and that correlates with the fact that this gene is actually transcribed. Later on, when we know that these genes are transcribed, these are also correlated with hypersensitive sites. So all this is saying is that we've got a situation where the same gene, well in this case different genes from the same region, are expressed at different times, and that DNase hypersensitivity correlates with the gene expression. So when the U gene is expressed, the U gene has a hypersensitive site, and when the alpha D and alpha A gene are expressed, they're correlated with a hypersensitive site. So that means that in these cells, or at this time in development, this chromatin is tight and this chromatin is loose. And then in this time in development, this chromatin is loose and this chromatin is tight. And in brain cells, where they don't express at all, it's all tight, because you don't need the genes to be expressed. So there's no hypersensitive sites in the brain, in the gene in the brain cells. So when you first look at that, that was pretty confusing, but hopefully it's okay. And we don't really need to talk much more about this. I've gone way longer than I need to. I was mainly interested in chromosome structure. All this is talking about is that the centromeres in cells have a function, and as we already know, during mitosis, the chromosomes get pulled to different cells by the spindle, and the centromere is the point of attachment of the spindle. That's basically all that is. And this is showing, this is just a quick experiment if you don't have a centromere. Oops. So this is a little fragment of a chromosome that doesn't have a centromere will get lost. It doesn't replicate normally. And then this is showing, I'm gonna have another PowerPoint all about telomeres, and you're doing an assignment on telomeres. But this is just showing that the telomere structure in various cell types is very similar. So this is looking at the ends of chromosomes. If you look at the sequence of that, you see a repeated sequence. So it's each of these things is showing. So this sequence would be repeated a bunch of times. And it's in lots of different things. And notice that in this little tetrahymena, and I'll give you a little lecture on tetrahymena in a couple minutes, has this sequence, and then this is what the human one looks like. So it's very similar. There's only one letter that's different between these two things. At least these are very, very, very different in terms of evolution. So tetrahymena is like a little ciliated protozoan. Humans are humans, and their telomeres are very, very similar. All right, so we're calling it good. So that's chromosome structure.